My name is Hussein Abkalo. I am a biotechnologist working at the International Livestock Research Institute in Nairobi. My primary duty is to generate live attenuated vaccine for African swine fever by employing the latest biotechnology tools like CRISPR-Cas9 gene editing technique. African swine fever is a viral disease of pigs that causes up to 100% mortality within 10 days of infection, depending on the genotype of the virus. The symptoms of disease include weight loss, loss of appetite, and hemorrhage in the infected pigs. The disease is usually passed on from infected pigs to non-infected pigs, either by contact or through consumption of meat or meat products. And also, if the pig comes into contact with equipments that have been exposed to the virus. The disease is present in 26 African countries, sub-Saharan African countries, and due to the lack of vaccines and therapeutics for this virus, the go-to system for controlling the disease through biosecurity, but that is also not applicable because the farmers are not in a position to incur the cost that comes with biosecurity. Because biosecurity is not really applicable in our context, the missing link, therefore, is to use vaccines, to generate vaccines expeditiously to control or prevent this disease. To this end, we are employing latest technology like CRISPR-Cas9 gene technology to accelerate the development of live attenuated vaccines that will help us to prevent and ultimately eradicate the disease. There are, of course, previous approaches to generating live attenuated vaccines. One is the conventional genetic approach, but the challenge with the conventional system is it takes a longer time to generate a single candidate. It's estimated to be around 6 months conservatively, but with CRISPR, we have shown in our labs that we can reduce that time to less than 2 months. And by doing so, we're able to generate multiple vaccine candidates. The other approach that has been used is cell passage. This is whereby you grow the virus continuously in a cell line until the virus is able to undergo some genetic modification and ultimately weakened. But the problem with this system is you are not able to control the part of the genome which gets modified. So with CRISPR technique, we can do precise and quick modification of target genes that have been shown to cause disease in pigs. One of the challenges we have is, lack of comprehensive understanding of technology, and people tend to confuse gene editing with GMO. And in terms of regulation, some authorities are bundling them together and controlling as one and the same thing. In my view, that should not be the case. We need to have a case-by-case, policy or framework that, governs gene editing. And the sooner we have this framework locally, I think the better for us to realize the benefits that comes with gene editing. My message to the farmers who are facing the challenges of African swine fever is that we have at our disposal latest technologies that will be able to generate multiple vaccine candidates within a short time. And we hope in the next few years we'll be able to have products in the market that will be able to control this disease.